Welcome to this explainer. Today we're tackling a massive, honestly long overdue scientific awakening about the human body. For decades, the medical establishment has relied on a foundational yet incredibly dangerous belief, treating women simply as smaller men. We're going to unpack a brilliant analysis today that consolidates 15 years of systems biology to show exactly why female biology requires its own entirely distinct structural framework. Let's get right into it.
To really understand where we are today, we first need to ask a question. Is a woman just a smaller man? Now, phrased like that, it sounds completely absurd, right? Yet for an entire century, biomedical research basically operated on this quiet assumption that the human body just defaults to the male body. And that female biology, well, it's just that exact same baseline, but with some pesky hormonal changes thrown in. Imagine treating a completely different operating system like it's just a slightly modified version of another. That is essentially what medicine has been doing.
Historically, researchers found it much easier to just filter out female variants. Menstrual cycles, pregnancies, menopause, these variables were messy. They introduced complications that were expensive and really time-consuming to study. So science opted for the path of least resistance and just used the male baseline. But modern biological reality tells a completely different story. Female physiology is not just some modulated male baseline. It is a profoundly distinct cellular system. So how did this actually become standard practice? Well, look at the history. Starting in the early 20th century, that implicit male model took root. Then, in 1977, the FDA issued guidance formalizing the exclusion of women of childbearing potential from early-phase clinical trials. That single decision hardened a flawed assumption into strict medical doctrine. By the 1990s, our entire medical infrastructure, we're talking diagnostic thresholds, blood reference ranges, and dosing tables, were overwhelmingly built for male bodies. But over the last 15 years, the dam finally broke. 3 massive discoveries in systems biology across genetics, immunology, and pharmacology have completely shattered this old medical doctrine. Let's walk through these breakthroughs to see exactly how the very foundation of medicine is shifting right beneath our feet. Let's kick things off with genetics. Every cell in a female body carries 2 X chromosomes, while males carry 1. To prevent a lethal double dose of X-linked genes, female mammals undergo what's called X-chromosome inactivation, essentially turning off one of those X chromosomes. Now, textbooks used to say this silencing was complete, done deal. But higher resolution science reveals a staggering reality. Roughly 15 to 23% of X-linked genes consistently escape this silencing in females. And this genetic escape, it has immense real-world consequences. Think about the people in your life. Chances are you know a woman managing an autoimmune condition. Many of the genes that escape inactivation are linked to our innate immune system. Because of this, women often mount much stronger immune responses, but it comes at a steep cost. The ratios for autoimmune diseases are absolutely staggering. Sjogren's syndrome impacts women 19-1 over men. Systemic lupus is 9-1. Hashimoto's is 7-1. The very reality of the female X chromosome directly drives these numbers. Genetics is just the first piece of the puzzle. This second breakthrough comes from reproductive immunology, and it's called microchimerism. Now, we all know pregnancy requires this sort of immunological truce, so a mother's body tolerates a growing fetus. But science recently discovered this truce is permanent. Fetal cells cross the placenta and they actually persist in the mother's body for decades. This means any woman who has carried a pregnancy is literally a permanent genetic chimera. How wild is that? And we aren't just talking about a few stray cells floating around. Living cells bearing another human's DNA have been documented taking up permanent residence inside every single major maternal organ system. The brain, the lungs, the heart, the liver, the bone marrow. These fetal cells integrate and live inside the mother's organs for the rest of her life. It is an absolutely breathtaking biological marvel. But the dual nature of these persistent cells is where it gets really fascinating. On one hand, they act as a trigger. The graft versus host dynamics of these non-self cells is actually a leading hypothesis for why autoimmune diseases cluster so heavily in middle-aged women, matching the timing of their accumulated microchimeric load. But on the other hand, they're healers. These exact same fetal cells have been shown to actively participate in maternal tissue repair, aiding in cardiac regeneration and even post-stroke recovery. So the crucial takeaway here brings us far away from those 20th century assumptions. The female body is not simply a smaller with hormones version of a man. Genetically and cellularly, we are looking at fundamentally different biological hardware. Our third and final major discovery lies in pharmacology, basically how the body processes drugs. A landmark 2020 analysis looked at 86 FDA-approved drugs. Only 10 showed male-biased pharmacokinetics, but a massive 76 of them were female-biased. Think about what that means the next time you or a loved one picked up a prescription. For the vast majority of these drugs, women experience elevated blood concentrations and take far longer to eliminate the drug from their systems compared to men taking the exact same pill. And the danger here is super stark. Of those female-biased pharmacokinetic patterns we just talked about, a staggering 96% are directly linked to higher adverse drug reactions in women. Because hepatic enzymes process drugs differently in female bodies, standard medical dosing protocols suddenly transform into a massive systemic risk. Which leads us to a really sobering conclusion, captured perfectly in the source material. Equal dose prescribing is not neutral. It overmedicates women by design. Every single time a prescription is written against a weight-based reference that ignores biological sex, it systematically overmedicates women. Medicine has known about these mismatched risks for over 2 decades, and yet equal-dose prescribing remains the default. So when you converge these massive discoveries in genetics, immunology, and pharmacology, you get a brand new framework of systems biology. Modern science now recognizes 4 pillars of female biology: the chromosomal baseline, epigenetic pacing, endocrinological modulation, and environmental intersection. They operate entirely independently, but they interact continuously to create the true female biological phenotype. To understand how this works in practice, let's break down pillars 1 and 3 using a really simple metaphor. Think of the chromosomal baseline as the body's underlying genetic hardware. It establishes networks completely independent of hormones. Meanwhile, the endocrinological modulation, the hormones, acts as the software. This software is continuously adjusting, updating, and sometimes even opposing that genetic hardware across life stages like puberty, pregnancy, and menopause. This distinct hardware-software architecture, it manifests in every organ system, altering everyday human experiences. In the vascular system, estrogen withdrawal at menopause rapidly accelerates blood vessel stiffening. In the liver, female metabolisms process chemicals at completely different baseline speeds. Musculoskeletally, a drop in estrogen accelerates both muscle loss and bone fracture risk simultaneously. And neurologically, female brain connectivity actually processes chronic pain in a completely different way than male architecture does. Okay, the evidence is undeniable. So how do we actually fix this? The source material outlines 4 urgent shifts for regulators and pharmaceutical companies. First up, mandatory sex disaggregated dose finding. Basically, stop assuming one dose fits all. Second, design programs that target the unique overlap of female comorbidities. Third, develop immunotherapies that actually account for those persistent fetal cells. And fourth, cycle phase pharmacokinetic protocols, meaning researchers must actually track where a woman is in her menstrual cycle when testing drug metabolism. It is well past time to stop treating biological sex as a statistical nuisance. The science of systems biology has spoken loudly and clearly. The female body is not, and never was, a smaller version of a male baseline. It is structurally, genetically, and cellularly unique. All of this leaves us with one vital, pressing question. Will the institutions that govern our healthcare finally adapt to this biological reality, or will they continue to risk women's lives by relying on a broken baseline?